Hi, everyone. I hope you're all revved up for the NF Core hackathon starting tomorrow. But it's time for bytesize now, and I'd like to begin by thanking the Chan Zuckerberg Initiative for funding NF Core events. I'd also like to thank those of you from the community that have actively been helping shape our outreach events recently and for making our material accessible. So we're back this week with a bytesize talk that will be focused on pipelines. And we have Daniel Straub, who is based at Cubic at the University of Tübingen in Germany, talking to us about the NF Core ampliseq pipeline. If you have any questions for Daniel, please either unmute yourself at the end of the talk or use the chat function, and I will read out your questions. Thanks very much for taking the time to present at bytesize, Daniel. Over to you.
Thanks, Renuka. So, as she already mentioned, I'm talking about NF Core ampliseq today. And to give an introduction, microbial communities are actually very important in our everyday life. So for example, we as humans, we have more bacterial cells than actually human cells. In agriculture, bacteria or microbial communities are also very important. They can be either beneficial or pathogenic. So it's also quite important here to know what's going on in order to improve. Also in the environment, microbial communities play major roles, like they are driving the global elemental cycles, meaning that the elements are turned over and recycled and can be actually used again by nature to build whatever it can build. So for example, here on the right side is a bacteria that are in the iron cycle, global cycle, or here on the lower figure, we also have a microbial community which is involved in removing arsenic from groundwater in Asia, where we also investigated the microbial community. And there are different ways to investigate microbial communities or mixed communities, mixed organisms at NF Core. We have, for example, a pipeline called NF Core Bacchus, which is for genome assembly of isolated bacteria. So from a microbial community, you can isolate a bacteria, extract their DNA, and then assemble its genome. Or from the whole community, can total community RNA be extracted, and then the meta-transcriptome can be investigated with NF Core RNA-seq. The DNA can be also extracted from the total microbial community to assemble the metagenome with the NF Core MAC pipeline, or a fragment of the community DNA can be amplified via PCR, polymerase chain reaction, and produce amplicons that are typically done for ribosomal RNA genes, rRNA genes. And that kind of data can be analyzed with NF Core ampliseq that I'm talking today about. But not only ribosomal RNA gene fragments can be amplified, but any other kind of gene or fragment that you might be interested in. However, ribosomal RNA gene amplification is typically used to investigate microbial communities and is the default of NF Core ampliseq. So I will focus now on that. So ribosomes are essential for the life. We know it. Ribosomes are translating the RNA in cells to peptides, to proteins, and that ribosomes contain proteins, but also non-translated RNA, and that is the ribosomal RNA, which is important for its structure. Prokaryotes have a specific set of ribosomal RNA, including the 16S rRNA, which is used mostly for investigating the taxonomic composition of microbial communities. Eukaryotes have different kinds of ribosomal RNA, and that is also used actually to investigate communities of those. So the 16S rRNA gene that encodes for that non-translated 16S rRNA, that is actually highly conserved, but it has regions that are variable and other regions that are actually very similar. So, and these variable regions of the 16S rRNA gene allow the discrimination of many bacterial taxa. And having conserved regions mean that we can use a PCR, primers for the PCR, to amplify that variable regions. So the problem of that is that there is an error accumulation during library preparation and sequencing. You can see that sample sequences here as 4 different dots in different sizes corresponding to their abundance have errors, meaning different kind of sequences when they are actually sequenced. So the sequencing reads have errors. So the traditional way of correcting this is producing so-called operational taxonomic units, OTUs. And those OTUs are basically lumping together all sequences that are similar to a specific threshold level. And for example, here it would then produce only 2 OTUs out of that 4 different original sample sequences. There are other methods which make so-called Amplicon sequencing variants, ASVs, and those methods try to correct the errors from the Amplicons back to the original sequences that are found in the samples. And first thing what I did is to compare those kind of methods. And what you can see here on the y-axis is the relative abundance of sequences in a reference data set. And the reference data set consists of 27 bacterial strains, which has 35 expected Amplicon sequences, because bacterial genomes can have several copies of that gene. And here you can see the reference, meaning this is how it should look like. There are high abundant sequences, but also very low abundant sequences. The color of the dots show if actually the sequence is a perfect match in green to the reference, in blue one off, so very similar, or in black, in a black cross, a sequence that is not expected. And you can see here that the OTU-based methods, here 4 columns for 4 different methods or settings, actually have quite a lot of those unexpected sequences. And they also in this region here do not find all the ones that we would expect, the green dots here. The ASV-based methods, here 2 different methods, do show a much reduced number of false positives. However, some of those sequences are also not detected for this method here. That is very strict. And I did that not only with 1 sample, but with 3 different samples, and with here the OTU-based methods and the ASV-based methods, not only for precision and sensitivity of finding the original sample sequences again, but also for the taxonomic classification and for a diversity index, the Shannon diversity. And here the green color shows the best methods, and the red one, the worst ones. And you can see that the ASV-based methods actually perform much better in the Amplicon sequencing analysis than the OTU-based methods. So coming from that, I wanted to produce a pipeline that is actually doing exactly what I need, and to use that kind of knowledge that I have accumulated by that benchmarking. And I wanted to produce a pipeline to analyze all kinds of Amplicon data with a wide range of input types, reference taxonomies, and downstream analysis. And we came up in 2018, then with the NF-Core ampliseq pipeline. In the end of 2018, we released version 1.00. Did some feature and maintenance updates. We also then published a paper about it, and actually continued afterwards in 2021 quite a lot with adding more features and to the pipeline. And now it is actually at a pretty good stage how I imagined in the beginning. So what is happening there in NF-Core ampliseq? And so this is here the whole picture. This is a bit complicated, so we will go step by step through. First of all, to start the pipeline, you would need such a command here. In red are the ones that are specific to that pipeline. First, there are different kinds of inputs that can be used. So either direct input of a folder containing all your reads or a sample sheet in a TSV. And that is what I describe here. So the TSV can contain, or it has to contain at least 2 columns with sample ID and forward reads, and 2 optional columns with reverse reads and sequencing run, because different sequencing runs of a sequencing machine has to treat it separately by the pipeline. And that sample sheet is then added with the input parameter. And using that kind of command will then end up in primer trimming, QC, quality filtering, and then also the inference of this Amplicon sequencing variants. For this, of course, you also need to add your primer sequences, because it's very important what kind of sequence was actually amplified. And depending on what kind of sequencing data from what kind of sequencing technology you have to also add additional parameters. So for Illumina paired-end sequencing, just default, you don't need to add anything else. For single-end, the single-end parameter for PacBio and for ion torrent technologies, you have to specify that. In special cases, actually Illumina paired-end ITS, so fungi community analyzers, you also should already set a parameter so that all the downstream analyzers will have sensible settings. What is the output of those kind of very basic analyzers is, so here in the results folder, in a subfolder, data will be that file, and that file here contains already most of the information that you will need. It contains, for each of the different samples, the sequences that it found and also the quantification. So it simply counts how many of the sequences in there will originate from that original 16S sequence in your sample. There is also a handy overall summary for all the read numbers that were for each of the samples processed and then actually ended up in the table. And if those numbers do not look as expected, then this is a very good starting point to troubleshoot. The next step of the pipeline is to also classify the sequences that we have now produced, meaning that we give it a name, that we give it a taxonomic name. And this is by default done with the Data2 software as well, but we also have an alternative way to do this with Shime2, which is also a very popular program in the area. And by default, it is, as I said, using the data algorithm with the Silver 138 taxonomy. So we have here a range of reference taxonomy databases that can be used. And that is the Silver, the RDP, the GTDB database that are all for bacteria, so for 16S rRNA gene Amplicon analysis, the Unite database for fungi analysis, ITS, internal transcribed spacers, and the PR2 database for eukaryotes with the 16S rRNA gene Amplicons. You can also actually taxonomically classify any kind of sequences that you have produced probably already by using -- input and provide your FASTA, which needs to have then a FASTA extension. Following, there will be taxonomic filtering, and by default, only mitochondria and chloroplasts will be filtered out from the following tables, because those are typically off targets of the 16S rRNA gene Amplicon sequencing that should be actually removed. And the output of this taxonomic classification is again a table, a TSV, that will list all the sequences that were discovered, and also add here the taxonomic levels and their classifications with confidence value. And finally, there is also downstream analysis, but this downstream analysis is only provided when the optional parameter metadata is pointing actually to a tab-separated metadata sheet. And then automatically appropriate metadata columns will be chosen, and used for visualization of a bar plot, as you can see here. Those are actually HTML files that are then interactive, so you can choose colors, you can choose different kinds of taxonomic levels. I chose here a very high one just to show you, for example, here with the test data set that we are typically running, here with triplicates, that you can see already patterns of different kind of data. Then additionally, there will be a differential abundance analysis with a program called Amplicon, and that produces, for example, such a volcano plot, also interactive. This red dot here, which is identified as a significant different abundant between the different treatments, would be, for example, a Burkholderia bacteria. Then also alpha and beta diversity indices are produced. Alpha diversity is a measure of each of the samples, how diverse the sequences in there are. And you can see that here for this test data set, we can see, well, it's a bit small, that here the groundwater has actually, the samples originating from groundwater have the lowest Shannon diversity index, but the sediment samples here have actually the highest one. To all of these comparisons will be, of course, also statistical analysis provided in the output of the pipeline. Beta diversity plots then show the difference in PCOA plots. And what you can see here is that the replicates for the different kind of sample sources, groundwater, river water, sediment, and soil here in our example data set are nicely clustering and well separated from the others. Additionally, there will be quality control figures like alpha rarefaction curves, and that curves basically show you if you have a sequence with sufficient output your samples. So as long as those curves will flatten out, you know that you have sufficient sequencing data produced for that kind of samples. All that results can be actually also found on the NFCore website in ampliseq results, and you can have a look what is actually produced, how does it look like, and there's also extensive documentation what each of those plots mean and what you can actually use it for. So that plots, of course, cannot show all kinds of complicated experimental setup, but they are very helpful for getting a first view on the data, and also as sort of a quality control of what you are producing here also makes sense. With this, I want to finish, and I want to thank you for your attention, my colleagues at Cubic, who also were involved in the development of the pipeline, and of course, especially also Daniel Lundin, who helped a lot, and the University of Tübingen Microbial Ecology and Geomicrobiology, who actually produced a lot of data that I was then able to analyze and with this pipeline and produce it as good as possible.
So thanks very much for that very clear and really insightful talk, Daniel. Are there any questions? If anyone in the audience has questions, you can unmute yourself or even add questions to the chat and I can read them out. Okay, I don't see anything coming up. So that was extremely clear. So I guess that's why people don't have questions. But if anything crops up anywhere, you know where to reach Daniel. You can message him on and continue the discussion on the bytesize channel. And thanks again for joining. We will be back next week with another pipeline focused talk. Oh, we have a comment here. Okay, there's a question. So this person thanks you for the presentation and asks what type of license is associated with this kind of tool, example, the ampliseq pipeline.
Yeah, so this is a CC BY license. So like I think all the NFCore pipelines have. And so there are actually no restrictions whatsoever.
I hope that answers your question. Yes, thank you. Okay. So as I said, we will be back with another pipeline-focused bite-sized talk next week, where we will have Payam Emami, who will be presenting the Metabo Igniter pipeline. But before that, I hope to be able to see some of you in sunny Gathertown starting tomorrow at our hackathon. And take care, everyone. Thanks again for joining.